Welcome today. Today, Dawn McSwain will be defending her Master's of Science in the Tropical Conservation Biology and Environmental Science graduate program at the University of Hawaii at Hilo. So just a little information about Dawn before we get this party started. Dawn joined TCBES in 2019. Before that, she earned a Bachelor's of Science in Biology from Eastern Washington University. Both at Eastern Washington University and more recently at Hilo, Dawn has taught undergraduate courses in Introduction to Biology and Oceanography as a TA and more recently as a lecturer. She's also mentored several undergraduate students in research projects. Dawn has also been involved in several research projects, including those focused on marine mammals and text analytics. She has also volunteered as a sea turtle stranding response team, the Marine Mammal Center, and Washington Department of Fish and Wildlife. With all of this being said, it is my great pleasure to introduce you to Dawn and to turn the floor over to her to tell you a little bit more about her master's research project. Dawn, take it away.
Thank you, Tracy. Hi, everyone. I hope you're all doing well. I'm going to talk to you today about my thesis project, Mixotrophic plasticity in corals, a meta-analysis using text analytic tools. I would like to say a little shout out to my nephew, Noah, who asked my brother recently what a biologist does. So here, little man, this is what a biologist does. So, coral reefs are rapidly declining worldwide at an unprecedented rate. Stressors such as thermal, acidification, sedimentation, and other anthropogenic pollution are responsible for their decline. It is currently predicted if global temperatures exceed a 1.5 degrees Celsius rise, then we can expect to lose anywhere from 70 to 90% of our world's reefs. Bleaching has been identified as a primary threat to corals. Now, historically, short-term bleaching events were not uncommon. Here, this infographic shows a healthy coral at ambient temperatures. When temperatures rise, then the coral becomes stressed. When temperatures continue to increase or stay too warm for too long, then corals will expel their algal symbionts, and this is what we call bleaching. A bleached coral is not a dead coral yet. If temperatures cool quickly enough, then corals can regain their symbiotic algae and become healthy again. However, we went from mass bleaching events happening every 25 to 30 years to every 5.9 years as of 2006, with yearly mass bleaching events predicted on the horizon. Combine this with other stressors, and the future of coral reefs is of great concern. Studies have found corals to be able to resist or rebound from single stressors, but have harder time when faced with more than one at a time. Kind of like a grad student, we can handle stressors one at a time, combine them all, and we kind of shut down and start crying. How corals that are able to rebound and survive repeat and prolonged bleaching events is still relatively unknown. By definition, scleractinian or hard corals are considered mixotrophic. They utilize both auto and heterotrophy to acquire nutrients, and I'll define each of these pathways in just a moment. However, a coral is typically categorized by whichever one they use primarily, and most corals are considered autotrophic. We're starting to realize that some corals can switch, or what is called trophic plasticity, between the two pathways. Like I mentioned earlier, corals are generally considered autotrophic. This means that they rely primarily on the nutrients derived from their photosynthetic algae that resides in the tissue, like the green we see here in this close-up of a coral polyp. This is the famous symbiosis we always hear about. Zooxanthellae, or symbionts, as I'll be referring to them from now on, provide nutrients to the coral in exchange for a safe place to live. When a coral is bleached, it's because the temperature became too warm for the symbionts, causing them to be expelled from the coral host, and then the coral is left without its main source of food. This is why it's bleached. The algae provides its vibrant color. Without that primary source of food, the corals struggle to survive. The other way scleractinian corals gain nutrients is through heterotrophy, or feeding. Here in this video, graciously provided by the Merhaver Lab at the Caribbean Marine Biological Institute, we can see a 1-month-old Dendrydra cylindris feeding on what most likely is zooplankton. It's extruding its polyps and it's pulling in to eat that food. Even though autotrophy can provide over 100% of a coral's metabolic needs, it's generally lacking in sufficient quantities of nitrogen and phosphorus, important nutrients needed for tissue growth. However, this is generally considered a secondary source of nutrition, anywhere from 15 to 35% of the overall coral budget. But if a coral can successfully switch to rely solely on heterotrophy when bleached, feeding has been found to provide up to 100% of a coral's nutritional needs. So this is interesting. Not only can heterotrophy provide more nutritionally robust energy, but can support a coral when bleached. Now, even though all scleractinine corals are considered mixotrophic, corals that exhibit trophic plasticity, or the ability to switch from primarily auto to primarily heterotrophy, are also considered mixotrophic. These corals are thought to be able to switch up their trophic strategy depending on their needs. So what do we know about heterotrophy? We've known about it and its necessity for scleractinine corals since the early 1930s, but overall, the extent of the benefits that this trophic strategy provides is understudied. What we do know that limits heterotrophy is resource availability, geographic location, and the severity of a given bleaching event. The last major review of heterotrophy in scleractinine corals was done in 2009, and it illustrated the various sources of food available to a coral and how nutrients gained through feeding can also have a positive effect on their symbiont's productivity and density. Generally, though, heterotrophy is considered species specific. There are still many unknowns, particularly which species can do this, and can heterotrophy, or the ability to switch, be beneficial long term. Since 2009, we've seen the impacts of climate change increase dramatically. We've also seen increases in coastal development, agricultural enterprises, and marine infrastructure that have added additional stressors such as sedimentation, excess nutrient runoff like we see in Pu'uko, and physical damage. Nearly 14% of coral cover was lost from 2009 to 2018. So since then, what has the literature found since that last major review in 2009? Is heterotrophy or mixotrophy a viable strategy for our corals to resist stress? I wanted to dive into the literature about coral heterotrophy and mixotrophy since 2009, but going through all of that literature can be daunting. I was thinking, is there a way to help me sort through all of this literature faster? This is where text analytics comes in. Text analytics is a tool that can help analyze large data sets quickly. It's typically used in the business world by companies such as Google or Yelp to automatically read thousands of reviews, sort them, and find important information to better their companies. Since it's automated, it allows for a more objective exploration of the data and can highlight patterns potentially not easily discernible by the human eye. The tool I'm interested in particular is topic modeling. Topic modeling can take an entire data set of text and sort it into major topics or themes that represent that data. This tool was found to be useful in other fields, so I wanted to see if I could find it to be useful for this field. So why does this matter? Oh, sorry. So why does this matter? Heterotrophy, do we know if heterotrophy is beneficial for all? Is it species specific? Can we find some patterns there? Could text analytics help improve the speed and accuracy of meta-analyses for other research fields? And with that, were we able to find if this is helpful and text analytics could help? This could help better tailor research goals, conservation efforts, and management plans because we can see what we do and don't know about different corals. And ultimately, as outplanting is becoming a more sought after active restoration option, knowing which corals can and cannot do this will help better those. So using the last 10 years of coral heterotrophic literature, I wanted to answer a few questions. First, can text analytic tools expedite and improve the meta-analysis process? 2, do certain coral genera display particular trophic modes over others? 3, what trophic modes are more successful than other surviving stress events? And 4, do coral trophic modes display any regional differences? The way I approached this study was twofold. First, I wanted to apply topic modeling tools to my data set and see how that compared if I conducted a traditional meta-analysis, meaning I read through and analyzed all of those publications myself. I'll walk you through the text analytics methods in this flowchart that I adapted from Amato et al. So to start, I searched all of the online collections I had access to, namely the Hilo online repository and Google Scholar, searching for all publications with from 2009 to the time December 2020 to publications relating to coral heterotrophy. I use the words corals, coral reefs, heterotrophy, and limited to those written in English and referring to heterotrophy in the coral animal. I briefly scanned all of the publications to make sure, the abstract, to make sure that they match that criteria, downloaded them all in PDF form. From here, I applied text mining tools. So this is where within Python, you can have the text mining tools extract all of the text. So everything from the introduction through the discussion into a single data set, applied some pre-processing steps just to clean up the text, make it easier to analyze. Now move into vectorization. Here we can look at have data visualization, we can look at word counts or create word clouds where this represents frequently occurring words. Now moving into topic modeling. Specifically, I used latent Dirichlet allocation, or what I'll be referring to as LDA topic modeling. Reason I used LDA topic modeling, it's one of the most popular ones, and it assumes there are underlying themes or topics to your data set. Here, moving on to 3D structure. This is a term, topic, article. This is where the process takes all of the words from the text, puts them in what's called a bag of words, and starts making associations with them, tries to find words that are meaningful and significant, and then this is how the topics are formed. You tell it how many topics to sort it into, and from that point, once the topics are formed, the documents, all the applications are sorted into its corresponding topic or the one it's most representative of. And here we can glean some insights into coral heterotrophic research. All right, so this first topic modeling attempt, I'll call it the beta testing. So I searched online, I found 69 total publications between 2009 and December 2020. I put them into Python, and I wanted to see how many topics to sort the data set into. I implemented coherence score, particularly CV coherence measure, and these help distinguish between topics that are semantically interpretable and ones that are just nonsense, trying to determine between what the computer might see and what is actually a topic. Created this word cloud here. So here we're seeing the 100 most common words within the data set. And this size directly relates to how often it occurs. The bigger it is, the more you see it. The color scheme doesn't have any association. It's just a nice, pretty visualization. Bleach, overwhelmingly, was the most frequently occurring word, which makes sense. Bleaching is a big deal. We also see carbon. That's the main source of energy. Makes sense. However, when I got to the point of looking at the publications into the topics, because I read all of them, I wanted to make sure it made sense. I realized that they didn't, those publications within each topic didn't really have anything to do with each other. So read through them again, maybe seeing if there was something the computer saw that I didn't, and I realized that using the whole body of text from the publications overwhelmed the model. Thinking back to the traditional uses of topic modeling, reviews are generally a short amount of text. There's thousands of documents, or can be, but a little text each. So I got to thinking, maybe I should just use the abstract text because that should have all the pertinent information about the study in a little blurb. So before I tried this again, just using the abstract text, I noticed that this word feeding kept coming up in the topic formations, but it wasn't in the word cloud. And I got to thinking, maybe I should do another search. 69 publications doesn't seem like a lot. Maybe I might have missed some. And so I added feeding onto that original search string of coral reefs, heterotrophy, heterotroph, and found an additional 12 publications. A little interesting because heterotrophy just means feeding, but found that this word feeding was very important in helping to form and inform our results. All right, so now I'm using the abstract text only. We'll call this topic modeling 2.0. So I put it all through the same process. And now I have all 81 publications relating to heterotrophy. Generated a new word cloud. Here we can see the word feed was important. It's now one of the biggest words here. Still bleach. And when I applied these coherence measures, this is what I used. So here we have number of topics on the X, coherence score on the Y, and you're looking for the highest coherence score before it peaks for the optimal number of topics to sort the data set into. This was 8, so 8 topics to sort that first topic model into. I remember I'd read all of the articles and I saw that within those 81, 66 of them explicitly investigated trophic pathways within a coral. So I was wondering if I created a second topic model from this manually curated data set, would those topics change? When I did that, similar word cloud with the second topic model with fewer publications, 7 topics was ideal. All right, so remember I said I also did a meta-analysis on top of this. So I want to walk through some of those meta-analysis methods. Using those 66 publications, I read through each of them and collected species and genera information, the location of where the coral was sourced, the trophic mode determined by the study, was it stressed, and did it survive that stress? The 3 trophic modes were autotrophic, heterotrophic, or mixotrophic. Even though scleractinian corals are all mixotrophic by definition, for my study, I referred to a coral as being mixotrophic if we saw it, or if the study found it to switch from one to the other. And so autotrophic is primarily relying on their photosynthetic symbionts, and heterotrophy was them relying solely on feeding, pulling food out of the water column. Types of stress were thermal, turbidity, increased food, acidification, sedimentation, high-low radiance, natural disasters, and good old-fashioned anthropogenic pollution. All right, I'm gonna go back for the topic modeling, the first one. So all publications, 8 major topics. Now, I had mentioned topic words before. When the model outputs the topics, this is what the computer is saying is a topic, and it's showing the top 10 most representative words. For the computer, it goes, here you go, this is a topic. So fatty acid, species, feed, trophic, host, heterotrophy, high source, zooxanthellae. That is what the computer told me is topic number 1. At this point, I have to go through and assign a meaning or name. And with this process, it's assuming you have underlying knowledge of the field. So I went through and bolded all of the words that were unique, meaning that I didn't see any of these bold words anywhere else throughout these topic words. And for me, I had read all of the publications, so I knew which ones were in each topic, and I assigned a name this way. So for that first one, I read off that list of words, fatty acids as a measure of heterotrophy in corals. I wanted to see if you didn't know all of the publications, you hadn't read them, could you still make meaningful and accurate names and meanings for each of these topics? So I contacted an expert in the field, Dr. Chris Wall, explained the premise and gave him this list of words in the same format, and asked him if he would be able to name this topic, and if we would come to similar conclusions. So he didn't know any of the publications, gave him this, and beyond semantics, our interpretations were nearly identical. So, okay, you can name these topics without having already read the articles, or you can also name them that way. When I looked at the distribution of publications by topic, so here we have topic number on the X, frequency of publications on the Y. For this first topic model, so this was all publications relating to heterotrophy, we found that topic number 5, heterotrophic strategies improve coral health when thermally stressed, was the most popular or most studied topic. And alternatively, topic 2, turbid coastal environments influence on energy acquisition, was the least studied. This makes sense. Bleaching is a thermal stress bleaching. It's a big concern. We're seeing it happen more often. However, we have a lot of research showing how coastal influences have a negative impact on coral reef systems. So it was interesting to see that there was the least amount of studies looking at how those stressors affect the trophic pathway of a coral. Okay, so topic model number 2. This was the refined data set, so only 66 publications, 7 major topics. Again, the computer went, here you go, here are your topics. I named them in the similar fashion and also had Dr. Chris Wall try to name them without seeing the publications. We came to essentially the same results. Distribution of this topic, so topic number on the X, frequency of publications per topic on the Y. Topics 1, 5, 6, and 7 were evenly distributed here with the most publications per those topics. Again, now it's topic number 3, coastal influence on trophic strategy, was the least studied topic. So what could I get from this? This first topic model showed how bleaching was the overall most studied and coastal stressors were the least, really highlighted that information. Now, being able to look at all of these publications by topic, it was able to illuminate key information. There were founding instances of where heterotrophy could actually be a detriment to a coral in some cases. And even if a coral can successfully switch to feeding when needed, if the right kind of food isn't available for the coral, then it doesn't really work for them. We're also seeing plankton, one of their main food sources, the distributions and compositions of them being influenced by climate change as well. And different corals preferentially feed on different types and sizes of plankton. So it really helped illuminate some of this. For the second topic model, what I found very interesting and really cool was that with the first model, this was the bleaching topic and this was the lipids topic. In topic number 2, those 2 topics were combined in topic number 5. So when I thought that was interesting, so looking at these publications, what it went from was showing how bleaching, or success surviving a bleaching event, was directly influenced by the lipid stores. So not only could a coral, if a coral switched to relying on feeding, it also needed to have these ample lipid stores prior to the event, or prior to the bleaching event, in order to survive through it. And also how certain types of lipids were important, how heterotrophy is the major contributor to, it's thought to be the major contributor to those stores. Again, highlighting how coastal stressors are understudied when it comes to how it affects a coral's trophic pathway. And the last and really interesting part was how you can refine the searches with these topic models in order to find new information. So I had manually curated the second set. I knew which publications I wanted to apply a second topic modeling effort to. But what you could do is you can take each of these topics, all the publications with each one, and apply the topic modeling process again, and seeing how the topics switch up and how, and highlighting maybe new important information throughout here. So it was really cool to see. And now, going and reading all the publications per topic, you know that they're related to each other, and it makes it easier to get through. Now, 81 publications isn't a lot. That's still a lot of literature to get through. But what if you had more? You now have more bite-sized chunks to get through because you're still going to need to read them at some point. The computer isn't going to completely erase the human influence, and we need to make sense of this. But it helps to categorize it and make it easier to work through. I found this to be really helpful when answering the questions I had for my meta-analysis. All right, like I just said, the meta-analysis, so I did a separate manual analysis of literature to sort of remind you what those questions were using the last 10 years of research. 1, do certain coral genera display particular trophic modes over others? 2, what trophic modes are more successful than other surviving stress events? And 3, do coral trophic modes display regional differences? Looking at the frequency of the different coral genera studied, now I had to back it up to the genus level because there were some coral species that were only studied once. I just wanted to see what was the most studied. Here we have frequency on the X, coral genus on the Y, and the overall most studied was Porites, followed by Acropora, Stylophora, and Posilopora genera. When I look at these genera in relation to trophic modes, here we have the results of my chi-square analysis. We have, this is a balloon plot. How you read this is there's coral genera on the Y, trophic mode on the X, auto, hetero, mixotrophic, and test statistics down here. If you look at the intersection of the two variables, you're looking at the size of the point or the balloon. And the larger the balloon, that's showing that that's happening more. And I want to really point out to you how Acropora, Stylophora, Porites, and Posilopora corals were most likely to be mixotrophic. They found they were able to switch to feeding when needed. Looking at my second question relating trophic modes to success surviving a stress event, another chi-square analysis. Now this time this is in a mosaic platform. You read it a similar way. You have the trophic mode along the top X, auto for autotrophic, het for heterotrophic, and mix for mixotrophic. And you have stress success categorized as yes or no. You look at the intersection of the two variables, and what you're looking for is size. So overall, I want to point out mixotrophic corals overwhelmingly were more likely to survive a stress event than the other two. However, I want to focus for a moment on the autotrophic corals. So even though mixotrophy was way more successful at surviving a stress event, within just the autotrophic corals studied, it's about a 50-50 split between surviving and not. So they can't be completely ruled out, and we should see what is making these corals still successful, even though mixotrophy has been found to be more. All right, for my last question, I was looking at regional differences. So first, again, I wanted to see what regions of the world were the studies happening. So frequency on the X, geographic region on the Y, and the most studies were conducted in the Red Sea, followed by the Central Pacific. Again, relating this to the geographic region to trophic mode, so region on the Y, trophic mode on the X. This is another chi-square analysis in balloon plot form, looking at the intersections of the two variables, and really want to highlight how the Red Sea found the most mixotrophic corals than other regions, but we can also see that's followed by the Central Pacific and Australia. Not only are we seeing these areas the most studied, they're more likely to have mixotrophic corals. So that first question looking at trophic differences between coral genera, we were seeing more genera-specific trophic modes, and now we're seeing regional specific differences in trophic modes. I found this interesting. I looked within the Posilopora genus and found 3 studies that specifically looked at Posilopora verrucosa, And these studies found it to be autotrophic in the Red Sea, mixotrophic in the Central Pacific, and heterotrophic in the Indian Ocean region. So not only are we seeing species or genera specific differences, maybe we're also seeing regional specific. The areas influence how these corals respond. Right, so comparing the topic modeling to the meta-analysis process, how well did it work? First, I'll talk about what worked. We're able to generate meaningful and accurate topics using abstract text that highlighted the dominant focus of the research as well as knowledge gaps. Really highlighted the importance of the word feeding just in looking for publications. And this was interesting because heterotrophy just means feeding. It made sense to use those additional search terms that seemed most logical, but really adding this additional term will populate more because 12 publications, that's a lot of research we would have missed out on. And now the topic modeling process, you have distinct, meaningful topics for future review. You know what the focus, where the focus has been, and can now relate them, reading through those publications in little bite-sized pieces to highlight and find important information, as well as doing additional topic models on maybe a refined data set, whether you curated it yourself or you did it on one of the topics and was able to pull out really important information. So what we learned. Initially, the text extraction process was a little difficult because there's variation in the publishing templates. The order in which the different sections occur, whether they're in all caps or not, there's hard, distinct boxes on the page, colors, or if there's hyphens in the words. That was the big one. Instead of text wrapping, if there was a hyphen, the pre-processing removes those symbols. And so now the word is split in two and misrepresented. For this study, I just copied and pasted the abstract text into a single file. That was relatively quick. I only had 81 publications, but trying to do that with a larger data set would be pretty tedious and kind of defeat the purpose of trying to speed up the process. So either finding additional text mining tools that have better success with all that variation, or coming to agreed upon template across journals would help facilitate that process. And I found that using the whole body of text overwhelmed the modeling effort. So, but with doing that, wasn't able to pull out some of that information, that specific information, because sometimes not all of the study species are included in the abstract text or regions. So there's some of that disconnect there. The other big issue is that there's too many words for the same concept. We're already seeing how feeding played a role, even though it's the same thing as heterotrophy. Also, when I was doing my manual analysis, the studies didn't refer to the corals in the same way. So especially when I was trying to determine the trophic mode of the coral, sometimes they said, oh yeah, relying on heterotrophy, or switched, displaying mixotrophy, or they might have said just increased feeding, or we found the isotopic analysis to be carbon depleted, nitrogen enriched, a way of looking at nutritional sources. So coming to agreed upon terms, especially just with the word mixotrophy itself. Maybe standardization of referring to scleractinian corals as polytrophic, and then referring to mixotrophy as the ability to switch back and forth. Overall, though, I found this modeling process to be an incredible complement to the traditional manual review by having the topics pre-sorted, all the literature, and being able to look at how the publications spread that way makes it a lot easier to get through. You know, reading through the literature can be daunting, but having this process where you can put the publications in, have the computer sort it for you, made it a lot easier and really will facilitate the process. So moving the field forward, Stable isotopes came up a lot throughout the topic modeling efforts and the meta-analysis, the manual process. That is the main way that trophic strategies are measured in corals, typically using delta 13 carbon and delta 15 nitrogen. Most of the time in combination, but sometimes one is preferentially chosen over the other. There is a train of thought that maybe it's regional differences or that environmental parameters are different, and that's why one might be seen to be better than the other across different studies. So if that is to be true, standardization of using both might be warranted in order to account for any of that variation. This also leads into the next point of fatty acid or lipid markers. Even using stable isotope analysis, it can be hard to differentiate the source of food because there is some exchange between the coral host and the symbiont. So trying to figure out where it came from can be a little difficult. There are studies that are finding that there are specific fatty acid markers that are better able to help differentiate between those. And so looking more into those various markers and implementing that more would be a great way to help delineate that. And lastly, the need for widespread monitoring. We've seen with a lot of these big conferences and reports, status of the world's reefs, that we're generally lacking in monitoring. And here we're seeing some areas focus more than others. So this is a call to help try to expand and improve upon what we know of our world's reefs. This leads directly into research and management tools. We have incredibly useful databases like the Coral Traits Database or World Marine Organization or Marine Organisms. Creating a database of what corals are doing as far as trophic modes would be really helpful. Here I was able to map using Google My Maps all of the studies, corals. So this time I could actually use species because I wasn't doing any stats. And there's a point for each of them on the map, color-coded by their trophic mode, blue for mixotrophic, yellow for auto, green for heterotrophic. If you zoom in and click on one of the points, it pulls up information like this. It has the species, the mode, the location, was it stressed, what type of stress, was it successful, and a link to that publication. During the process of my own project, access to this map was requested to help make better or choose better study species. So this is a great start. There's a lot of information on here. However, there's lots of areas that are lacking. Even just within the Hawaiian Islands, the only information I have over the last 10 years is from Kaneohe Bay. We are even seeing morphological differences between the corals on Oahu versus here and through the rest of the islands. So expanding those efforts, finding out what our corals are doing here and everywhere will help to better tailor research efforts and also management plans. Knowing what our corals are doing and having the most information about them, we're going to be able to create more meaningful action plans as well as restoration efforts. So in conclusion, 1, topic modeling tools are successful in categorizing the last 10 years of coral heterotrophic research and have been found to be a great complement to the meta-analysis process. 2, mixotrophy overall was found to be a better survival strategy for corals. 3, trophic differences. We did find trophic differences between coral genera and also regional differences between trophic modes. And lastly, as the Prime Minister of Barbados, Mia Mottley, so eloquently put it at the UN Climate Change Conference last week, national solutions to global problems do not work. The task of learning trophic strategies of individual corals around the world is daunting at best. The only way to accomplish this goal is to engage local community members and stakeholders to learn this information. There's a Hawaiian alele noio, or proverb that goes, ahoya hua'o ka mai'a ka la ho'oko ka'i, which means when a task is done together, no task is too big. This is a great opportunity to invest in local communities worldwide and get all hands on deck to study local corals. When the pandemic hit, lockdown caused many long-term data sets to be disrupted because researchers couldn't travel to the remote or distant study sites. Our study demonstrates the need to get rid of this practice of parachute science, where typically wealthy scientists drop into a remote, low-income area, conduct their research, then leave. Tasking local communities to find this information not only protects the viability of long-term data sets, but leads to more meaningful and robust science, since the ones conducting it are the most to benefit from it. Science is useless unless it directly benefits the local community. This is also a way to create more job opportunities and diversify the research field, as it is so desperately needed. While almost 14% of coral cover worldwide was lost between 2009 and 2018, we regained 2% of that coral cover lost between 2018 and 2019. Corals are still fighting. Now we need to fight along with them. I'd like to take a moment to thank Dr. Tracy Wigner, my wonderful advisor, for all of your help and guidance throughout this process, Dr. Supo Hong and Dr. John Burns, my wonderful committee that helped form this, and with all of their great insights and feedback to get the best possible results. Lisa Parr, who has been an outstanding and supportive mentor throughout my entire time here at Hilo, to Dr. Chris Wall for being so graciously willing to help out. My TCBES cohort, I love you. You wouldn't have been able to do this without you. And all of the faculty who took the time to talk to me whenever I needed and always there to lend a hand. The Marine Science Department for, again, all of your support and also helping fund me for the past 2 years. The Marine Mammal Lab for all of your wonderful support. You've gone through many practice runs of this presentation, and the Marhaver Lab for being so willing to share all of your adorable coral videos. And I couldn't end without thanking all of my family and friends. They say it takes a village to raise a child. It takes a village to raise and complete a master's degree. My wonderful husband, who has been there through everything and was so willing to up and move across the world for me to pursue this degree. My mom and my in-laws for always supporting me and helping out any way possible. My wonderful friends who have been through all of it with me, many iterations of this project and all of the others. And I wouldn't be able to leave without acknowledging the 3 best and fuzziest study buddies a girl could ask for: Professor Peaches, Captain Dusters, and Dr. Pepper. They've been there in my lap and next to me through reading everything, all of the analyses, and would be here in this project right now if he wasn't currently being locked out and in the other room. So, mahalo nui loa. Any questions?